Bueno, buenas tardes a todos. Voy a hablar en inglés, pero voy a agradecer primero en español la cálida presentación de Pablo. Pablo fue alumno mío, Marcelo fue alumno mío, muchos otros aquí también, levanten la mano. Eric fue químico, no fue alumno, pero fue docente y coordinador de la materia. O sea, hay un universo de gente querida y conocida acá. So I will switch to English. In fact, I think I'm a little bit like an intruder here because I'm not a plant molecular biologist. I am an animal molecular biologist. I always worked in animal cells, in human cells, but for the last 6 or 7 years, one of my students and postdocs started a project in plant alternative splicing. So, in my introduction, I will talk about alternative splicing, and I will give some examples of animal alternative splicing, and then I will switch to the main subject, which is light regulation of plant alternative splicing. So, the title I chose is Shedding Light on Alternative Splicing. As you will see, it's not only white light, because when I talk about the animal cells, I will talk about UV light. Okay, so I start my talk saying that there is a renewed interest in alternative splicing, but actually this renewed interest is 20 years long now. We know that it's more a rule than an exception. It is estimated to affect 95% of human genes and 65% of plant genes. It explains how a vast protein diversity can be achieved with a limited number of genes. And mutations that affect alternative splicing regulatory sequences that we call splicing enhancers and splicing silencers are a widespread source of human disease, mainly hereditary disease and cancer. But our lab has been working for the last 15 years in something that tells us that there is a coupling between alternative splicing and transcription and chromatin. So alternative splicing regulation not only depends on the interactions of splicing factors with our target sequences in the pre-messenger, but is coupled to RNA polymerase II transcription. And I will just show two extreme examples of the importance of this process. One is a very well-known textbook example. This is VSCAM gene in Drosophila. This gene has, 4 regions of alternative splicing, which can have 12 alternative exons here, 6 here, 33 here, and 2 here. And then the combination of all the possible mutually exclusive events gives rise to up to 38,000 different messenger variants. Of course, the generation of these variants, mechanism is known, is stochastic, but the function of these stochastic variants is selected and has a function in the development of the nervous system in the fly. But what always amazes me is that this is a philosophical problem, because the number of variants the fly can make from this gene is higher than the number of genes the fly has, because the fly has only 16,000 genes and it can make 38,000 different variants from a single gene. So the whole idea when we started to study molecular biology of one gene, one enzyme, one gene, one protein, one gene, one polypeptide, one gene, many polypeptides, is now we are overwhelmed about all the possibilities that exist that we didn't know before. So the second example is an animal example, is a particular alternative splicing that occurs in neurons in vampire bats. These animals are obligate blood feeders. They feed on blood, and they can sense infrared radiation. And the fact is, to sense this radiation, they have particular structures that are along at both sides of the nose. And the ion channels of the terminals in those structures are the same ion channels that all animals we use to sense heat, to sense noxious heat. But the difference is that in those terminals of the trigeminal ganglia, these channels are open at temperatures that are above 30 degrees instead of above 43 degrees. So there is a change in sensitivity to the temperature. And this change is very important because it's what allows these bats bats to feed. So this change is just performed using the same gene with a change in alternative splicing that removes the carboxylic terminal domain, and now the channel opens at lower temperature. So this is a nice example of tissue-specific and species-specific alternative splicing that gives rise to something that is, of course, selective. As Marcelo mentioned this afternoon, there is a whole set of regulatory events on alternative splicing. The classical ones that are highlighted here in red or brown are the post-transcriptional ones, in which RNA secondary structure, RNA sequences that, as I mentioned, are called enhancers or silencers, and splicing factor abundance and modifications, and even core splicing proteins, as Marcelo mentioned, can affect the outcome of alternative splicing. But then now we know that a series of events occurring at the chromatin level and at the transcription level are also important. For instance, we found that promoters and transcription factors are important for the result of alternative splicing. In 2009, there was an avalanche of papers indicating that nucleosome positioning, nucleosome preferentially positioned on exons, is also important for alternative splicing. There is recruitment of factors to histone marks that is important for alternative splicing, and there is also importance on the speed of RNA polymerase II in terms of splice site choice. And we focused for many years on the fact of the control of speed by RNA polymerase II, and we found that splicing and alternative splicing are coupled to Pol2 elongation, and this is a cartoon I always use, this movie, to show how a change in speed of elongation can affect splicing decisions. For instance, if we have an alternative exon that has a weak splice site and a strong splice site in competition, if Pol2 goes fast, then the strong site is used, outcompetes the weaker sites, and then that leads to exon skipping. But if Pol2 goes lower, we learn that there is recruitment of splicing factors to upstream instro to the weak site before the stronger site is even made. So in those conditions, then the exon will be included, and it doesn't matter the order of intron elimination. In fact, in the fibronectin exon 33 that we use as a model, the second intron is spliced first, and the first intron is spliced second. So the order of intron removal is not important. What is important is the slow Pol2 allows recruitment of splicing factors to the upstream intron first. So what can change Pol2 elongation in a physiological situation? So far, we studied this in animal cells, but there is evidence now that this process also occurs in plant cells, although this is not our evidence, it's not from our lab. So essentially, we thought that it could be changes in the Pol2 intrinsic activity, either phosphorylation of its carboxy terminal domain or association to elongation factors, or changes in the template chromatin structure that could limit or facilitate elongation. I will just show 3 examples from my lab showing this, and then I will go directly to the plant story. So, changes in template chromatin structure that limit or facilitate elongation. We had plenty of evidence for this. One of these models in which we were working is the NCAM exon 18 alternative splicing, and we found that the same gene in 2 different situations acquires different histone marks. In one case, upon neuron depolarization, or if we treat the cells with trichostatin A, which opens the chromatin and hyperacetylates the histones, we have a more open chromatin which allows for a faster Pol2 elongation, which leads to exon skipping. But the same gene, in a different situation during neuron differentiation, doesn't have this mark, and then the histones are methylated in lysine 9. This creates a more compact chromatin structure that creates a roadblock to Pol2 elongation, and that promotes exon inclusion. So we have the possibility of 2 different chromatin situations, which lead to 2 different alternative splicing results. So we call these alternative chromatin alternative splicing. Then another series of experiments in a different system show that we can create roadblocks to elongation inside the genes, in the gene bodies. And for that, we took advantage of what is called transcriptional gene silencing. It means transfecting cells with siRNAs that are targeting intronic sequences. And we know that this depends on the argonautic protein AGO1 that presents one of the two strands of the siRNA to the nascent pre-mRNA, and this base pairing creates or promotes the dimethylation of lysine 9 and the trimethylation of lysine 27, the recruitment of the heterochromatin protein HP1 alpha, also HP1 gamma also works, and then this creates a roadblock to elongation locally inside the gene that of course affects alternative splicing. And we show this method, this procedure for several alternative splicing exons for several genes. And the take-home message is that we shouldn't only look at histone marks that operate at the promoter level, but just pay attention to changes in chromatin that are intragenic, because they might not alter the rate of transcription, but they might alter the rate of elongation inside the gene, and then change the quality of the message instead of the quantity of the message. So the other possibility that I mentioned is the modulation of POL2 intrinsic activity. I will not go in detail in this, but the best model we've been studying in the lab is how DNA damage produced by UV radiation modulates alternative splicing. And to make the long story short, I just will tell you that when we irradiate the cells with UV light, there is a hyperphosphorylation of RNA polymerase II. This hyperphosphorylation slows down RNA polymerase II, and many alternative splicing events change, and this was confirmed by many labs now, and this change in some cases can go to higher exon inclusion. And some of these events that change are those in which the options are pro-apoptotic or anti-apoptotic isoforms of the messenger. And what happens is that those events which is upregulated after UV radiation is the pro-apoptotic, which means that the cell, in a way, is responding by promoting death instead of expanding the putative mutations that are being created by the UV radiation. So, so far the introduction, and actually I say, well, we go from UV light to white light, and to white light in plants. And the reason why we started to work in plants, as I mentioned, and it's related to the reason that Marcelo mentioned about the work with PRMT5, is because one graduate student, Ezequiel Petrillo, who we call Petri, when he was going to start his PhD, he came to me and said, Alberto, don't you want to work in plants? And I said, yes, it is like music to my ears, because I was trained as a botanist and I like plants, but it was difficult because we were not experienced and we didn't work with Arabidopsis, we didn't know even how to grow it. But he learned very quickly, thanks to the help of Marcelo Janowski and Pablo Sernan, and he learned quickly, but the experiments took very long, because working with plants, as you know, takes long. And the story I'm going to tell you, as I mentioned before, took almost 6 or 7 years to get published. Fortunately, it got published in the journal. But the fact is that Petri wanted to know whether there was an effect of light-dark conditions on alternative splicing. And we used an alternative splicing event of a gene that encodes an SR protein. SR proteins are proteins that regulate alternative splicing, and plants have many more SR protein variants than human cells. And this was a model in which there are 3 alternative splicing variants, mRNA3, mRNA2, and mRNA1, and only mRNA1 encodes the real protein, that is RS31. But these 3 variants exist and are not precursors. The precursor is a different transcript. So this is the light-dark protocol. He kept the seedlings for 1 or 2 weeks in constant light, and then he transferred them either to dark for different times or kept them in light. And you can see here that we are measuring the splicing index. The splicing index is just the ratio of the longest isoform divided by the total isoforms. So even though this is not the one that is generating the active protein, that's the way that the community now wants to measure splicing. Alternative splicing, when there is more than one isoform, is the ratio of the longest one divided by the total, the sum of the three. So in this splicing index, each black bar corresponds to the ratio to the splicing index in the dark, and the white bar to the splicing index in the light. And you can see that after 24 and even 48 hours in the dark, there is a huge difference in the splicing index between dark and light. And this difference is proportional to dark, to light, the way you want to see it. You see here decreasing light intensities, and proportional to the decreasing light intensity, we see an increase in the splicing index. And at the very beginning, we showed that this apparently was gene specific, because this is another splicing regulator, another messenger, RS2Z33, and there is no difference in the splicing index between the light and the dark conditions. So it seems to be something, at least in the first case, gene specific. So one of the things we learned very early is that the time needed to get the dark ratio was longer than the time needed to get the light ratio. So to get the dark value, we had to wait for 40 hours, 48 hours. But to get the light value, it was sufficient with 1 or 2 hours after taking the seedlings from dark to light. So there was something related to the signal that was not the same to generate or to destroy a signal in the dark than to generate or destroy a signal in the light. So the first, of course, thought was that light perception for photomorphogenesis and for things that are related to growth and to splicing should be related to sensory photoreceptors. So I'm shy about talking about this here because I'm not an expert in this, and you are the experts, you are the plant physiologists. But the fact is that, as you know, the candidates could be cryptochromes for phytochromes. And the first thing Petri did is just to use monochromatic light, to use LEDs with very, very monochromatic light. And the surprise was that the white light effect was observed both with blue light and red light. So it meant that either the photoreceptors were not involved, or both photoreceptors were involved, because we got the same effect with blue and light. But then he started to use mutants, and he used mutants in blue light reception, in red light reception, and the integration of both signals. I'm going to show only some of the mutants, but essentially, here you see the monochromatic effect. You see it's exactly the same as the white light. I repeat the same experiment. But in these that are integrators of the phytochrome and cryptochrome signals, we see the light and dark effect, no problem, even in the double mutant. And this is a cryptochrome mutant, and again we see the effect with white light and with blue light as indicating that the cryptochrome and the phytochrome were not involved. We have also phytochrome mutants. So whatever it is, it didn't seem to be the photoreceptors. In the meantime, we had to show that the changes we were observing were actually changes in splicing and not changes in mRNA degradation. This is what I asked this afternoon to Marcelo. And in this case, we used 3 different approaches. One approach is to treat the seedlings with actinomycin D. Actinomycin D inhibits transcription. So if we saw a change after inhibiting transcription, that could be due to differential mRNA degradation. But fortunately, we saw a change, the regular change, in the absence of actinomycin D, but in the presence of actinomycin D, the change was not observed. So it's not a change in the degradation. But that was not sufficient. We also used mutants, UPF mutants, that are mutants in the nonsense-mediated decay pathway. This pathway degrades messengers that contains premature stop codons. And this maybe is well studied in animal cells. Marcelo mentioned this afternoon that it's not always working in plants. But in any case, 2 UPF mutants behave like the wild type in response to light and dark. And finally, Micaela GODOY HERZ, who is continuing with the subject, grows protoplasts and overexpressed in the protoplasm by transformation a plasmid expressing the splicing factor that is U2F65, and she saw this splicing factor recognizes 3' splice sites, and since our alternative splicing is due to weaker 3' splice sites, if we overexpress the factor that the target site is weaker, we get a similar effect as the light effect. So these 3 lines, independent lines of evidence, convinced us and convinced the reviewers that we were looking at alternative splicing and not differential mRNA degradation. Another control we had to perform is to show that the change from light to dark was not due to the particular artificial protocol we were using 2 or 3 weeks in light and then changing to dark. So we had to show that the effect was observed in normal cycling plants. And actually the effect is observed, but only in short day cycles. In short day cycles, at the end of the day, we see a lower splicing index compared to the end of the night. And this is what we see. If we go to long day cycles, then we don't see the effects. Probably means that the night was not sufficient to abolish the effects of light or vice versa. One of the other things we had to rule out is that our effects were not due to circadian rhythms. And that is, again, the help of Marcelo. And in this case, what you see here is that this is the apparent time, the subjective time, and at the end of the day, we see the light and dark effect as we see it at the end of the subjective night. But the fact is, we see it in several mutants, several clock mutants, okay? So again, photoreceptors are not involved, and apparently this is also observed in clock mutants, which means that it's not a problem of circadian behavior. As you remember, I mentioned that the shorter isoform was the only one that gave rise to the active protein, to the real protein. So what is this alternative splicing mechanism serve to? And the fact is, it serves to downregulate RS31 messenger 1, which is the active one. You can see that in our light and dark protocols, the total levels of RS31 RNA don't change. But because of the change in splicing, we have a huge reduction in the amount of the active messenger. So I'm going to say that probably the physiological role, and we will see this in a way at the end, is to reduce the amount of the active messenger at night. And this is what happens, not because of the reduction of transcription, but because of a change in alternative splicing. So the question we had in front was the generality of the phenomenon, the mechanism, and the biological relevance. So, what about the generality? We collaborated with John Brown and in Dundee, and they had a panel of about 100 alternative splicing events, and we prepared RNA from the light and dark from our seedlings, and used that to challenge the panel. And you will see that in 61% of the alternative splicing events, we did not observe any change from light to dark, but in 39% we observed changes from light to dark. And interestingly, the majority of these events, 35% of the total, this change was not reverted by the addition of sugars, which means that the dark effect was not the consequence of sugar starvation. Because if we added sugar, we still saw the dark and light effect, mainly the dark effect. So what was nice is that this panel allowed us to identify 2 other robust alternative splicing events to work with. One of them was the actual U2EF65 alternative splicing, and the other one was another splicing factor, SR30, and you can see the changes from light to dark with these two other alternative splicing events. So now we have three to test our mechanism, which are RS31 with 3 messengers, U2A65 with 2 messengers, and SR30 with 2 messengers as well. So what about the mechanism? So far we knew that photoreceptors apparently were not involved. So one of the first clues that perhaps was due to the chloroplast, to the organelles, maybe mitochondrial chloroplast, but actually mitochondria don't sense light as far as I know, was that Petri inhibited organelle transcription with an antibiotic, with rifampicin. And when he did so, he prevented the effects of light. So inhibition of the organelle protein synthesis, in this case at the level of transcription, prevented the effect of light and dark. So this made us think that maybe the chloroplast was involved in this effect, which was consistent with the fact that we obtained similar results with a monochromatic red light and monochromatic blue light, that are the lights to which the chloroplast is sensitive. So one of the most beautiful experiments was to do this, to treat the seedlings with light and dark protocol, and then 2, 4, or 6 hours after the treatment to dissect the shoots from the roots, and then to extract RNA from the shoots and from the roots, and to see whether we observed the effect, the change in splicing. And you can see here that in the shoots, we observed the effect from the very beginning, 2 hours, 4 hours, 6 hours, which is light, dark, light, dark, light, dark. But in the roots, 2 hours, dissecting 2 hours after of treating the seedlings, we don't see any effect. And we start to see the effect at 4 hours, and we still see it at 6 hours. But then the control experiment is to dissect first the seedlings, and then to apply the light and dark protocol either to the shoots or to the roots. Leave for 2, 4, or 6 hours, and then extract the RNA from the dissected shoots and the dissected roots. And you can see here that in the shoots, you see the effect 2, 4, 6 hours, but in the roots, you never see the effect if you dissected them before. So that tells us that whatever it is, there is a signal that is generated in the green parts, because the green parts are needed to see the effect on splicing in the shoots, in the non-green parts. So we were very happy with this, but then the reviewers said that dissection is stressful, and it creates stress to the plants. So they say that we should do covering experiments. So people who already listened to my talk know that I always use this metaphor. But we actually did the experiment covering the shoots and the roots. And what you see here is the splicing index of RS31, but only in the roots. We are looking at the roots. So if we cover the whole seedling, of course, in the roots, we see a dark ratio, dark splicing index. But if we cover only the shoots, we see exactly the same, well, there is no difference here, no statistical difference, of ratio dark in the roots. But if we cover the roots and leave the shoots uncovered, then in the roots we see a light effect. So it's exactly the same conclusions as the dissection experiments, but without dissection. Then physiologists tell me that covering also is stressful. But there is another kind of stress, I suppose. So two different stresses cannot have the same influence on the main experiment. So if the chloroplast was the culprit, then we had to inhibit photosynthesis and see what happens. And the first thing was to use this VCMU that blocks the electron transfer from photosystem 2 to the plastoquinone pool. But in fact inhibits the whole chain. Of course, there is a reduction in the reduced NADPH, of course. And then you can see that DCMU mimics dark, because increasing concentration of DCMU in the RS31 alternate splicing event increase the bar towards the dark effect. And you can see the same with the other two alternative splicing events, U2F65 and SR30, and you can see that DCMU mimics dark even though the plants are being illuminated, are being exposed to light. But then Petri, and it was new for our group, and it's not new for the people who work in photosynthesis, decided to use another inhibitor, that is DBMIB. And DBMIB inhibits the flow of electrons and the reduction of cytochrome B6F and keeps the plastoquinone pool reduced. And it should inhibit the whole chain. So, in principle, if the effect of darkness was at the end of the chain, we should see exactly the same mimicking of darkness that we had seen with DCMU. But not. With DBMIB, we saw that in low light, DBMIB potentiates the effect of light. And in light and dark, DBMIB mimic light. So this is RS31, control light and dark. With DBMIB, then the dark bar is lower. And for the other two alternative splicing events, exactly the same. Control DBMIB, control DBMIB. So it means that another inhibitor of the whole thing, of the whole electron transfer chain, provoked a different result. So it mimicked light when DCMU mimicked darkness. So this, so far, led us to think that the reduced plastoquinone pool was the origin of the signal that started in the chloroplast and acted some way in the alternative splicing regulation in the nucleus. So about the biological relevance, I always say that this is the weakest link of the whole story. We collaborated with Andrea BARTA and with Maria KALYNA in Vienna, where Petri is now doing the second postdoc in their lab. And what we did with them is to create transgenic plants which overexpress messenger 1. Remember, messenger 1 is the one that goes down, is down-regulated in the dark. But also controls in which we use a mutant, an insertion mutant of the RS31 gene, but that keeps the alternative splicing event alive. It doesn't make the protein, but the splicing event is upstream of the transposon insertion. And then also the overexpression of the whole gene in another position with another promoter. So essentially, none of the transgenic overexpressor of messenger 1 or the mutant of the gene or the overexpressor of the whole gene had a conspicuous phenotype in terms of normal day-night cycles. The only thing it's important is that when we overexpress messenger 1, we overcame the reduction that naturally occurs in the dark. So this is the condition of overexpressing in the transgenic in messenger 1, so we don't see changes because we are saturating the system with messenger 1. And this seems to be the cause of the problems when we go away from the normal light and dark cycles and we go to extreme conditions. In extreme conditions, for instance, extended darkness or constant light, the one that suffers more is the one that overexpresses messenger 1. And those are aborted seedlings in which when we overexpress messenger 1, we overcome the alternative splicing regulation that we found that the purpose was to reduce messenger 1 during the night. When we overcome that, the seedlings suffer and they don't progress. And we think that for this messenger, the important thing is just to cycle during night and day and not to keep constant because it's not good. So essentially, the summary is that we found that light and dark conditions affect alternative splicing of several alternative splicing events, that the signal is probably triggered in the chloroplast by the reduced plastoquinone pool, and then when this plastoquinone pool is high, there is a signal whose nature we don't know that affects splicing in the nucleus of the shoot cells but also splicing the nucleus of the root cells. And we don't know the nature of the second signal. We suppose that it's not the same signal, that it's another one, that it's a different chemical nature. In fact, in the dark, because of the reduced plastoquinone pool, we don't have the effect on the alternative splicing in the nucleus. So the conclusions is that light regulates plant alternative splicing, that a retrograde signal triggered by the chloroplast reduced plastoquinone pool mediates the effect of light on nuclear alternative splicing, and the light effect is also observed in the roots when the communication with the photosynthetic tissues is not interrupted, suggesting that there is a signaling molecule that travels from the shoots to the roots. And I'd like to thank mainly PETRILLO, who is the hero of this story, and her follower, Micaela HERZ, who worked with Petri during the last period of his work, mainly trying to do the experiments to respond to the reviewers. And now she's doing her thesis, trying to see what conditions are important for the signal, mainly for the signal that goes from the shoots to the roots. And also I'd like to thank the collaborators that I mentioned already, Maria KALYNA and Andrea BARTA in Vienna, this is Spanish, John Brown and Craig Simpson in Dundee, and Marcelo and Pablo in Buenos Aires. I'd like to thank the other people in the lab. Many of the things they do, I didn't have time with the purpose to tell here because this is a planned meeting, but we work in different subjects, and actually there is no conflict in working in a single lab, in someone who works with seedlings and some other who works with mice brains or human cells in culture. We are biologists. So I like to thank these people, and also I don't know whether the funding was there. Thank you very much.
Questions?
Can I say hello to them? Hola.
Thank you very much for your class once again. And do you have any idea what does the protein look like in terms of sequence?
You mean
The product of the
It's an SR protein. It has serine arginine-rich regions, and it was described as a splicing regulator. And in fact, in the panel, there is a preference of the events that change are mostly related to RNA metabolism, either RNA processing or RNA transcription. So there is a bias, although I always say I don't believe really in the gene ontology studies. I think the gene ontology is just something to mention, it's not key. But the fact is that the splicing events that are affected by light and dark seem to be biased towards RNA metabolism.
Thank you, Alberto. It's an excellent talk. There's a plastoquinone oxidase that oxidases plastoquinone in the presence of oxygen. Do you think this protein could play a role in alternative splicing?
No, but I didn't know that there was this. This is outside the chain.
Yes.
It is another protein that takes the plastoquinone pool, reduced, and oxidases it.
With
well, I'm learning.
oxygen.
I'm learning. Did you have mutants of that?
There are mutants. I don't work with that.
No, I don't have any clue on what you are suggesting, but I'm thrilled about the suggestion. So if there is something that can reduce the plastoquinone pool. So we measure, which is a little bit, if you want, known that in light conditions, plastoquinones are reduced and in dark conditions are oxidized. But we measured with the people at the organic chemistry department the pool of plastoquinones in our protocol, and they change as predicted. But we never thought or used any mutant or any condition that could withdraw or could reduce downregulate, let's use the word, or the reduced plastoquinone by other methods that are not just dark or DBMIB increase.
Yes, it regulates the rate of electron transport. So.
Well, should we talk about it? You should talk.
So what happens under UV light? Because that gives a stress to the plastoquinone pool.
We are not working in UV light in plants, actually. We haven't done anything. There are experts in this country. We work in UV light just in animal cells. We haven't done any experiment UV in plants. I don't know. Yeah.
Bueno, mi pregunta en castellano. Nosotros trabajamos en estrés oxidativo y por ahí, no sé, se ha pensado en que podría ser algún efecto de especies activas del oxígeno.
Yes,
Es muy simple, ¿no? Pero a lo mejor con antioxidantes.
Yeah, she's asking whether we looked at ROS, reactive oxygen species, whether we either mediating or performing something on automatic splicing. Well, I remember Petri tried many mutants, and he also tried hydrogen peroxide, and apparently it wasn't involved. So the experiments I remember at the very beginning, he sort of scanned these tools to try to see whether there was something related to ROS, and he ruled them out. But I don't have exactly now the experiment. I remember he used hydrogen peroxide, and there was no difference.
Maybe.
But it's maybe, I mean, hydrogen peroxide may be very tough, I would say.
Okay. Or maybe superoxide de anion, or maybe hydroxyl radical.
Yeah, as I told you, I cannot tell you exactly the experiment, but that's something that was considered at the beginning of the project, and we really didn't have any conclusive evidence as to include it in the menu. Okay. So it was out of the menu.
Thank
you. Regarding the biological relevance of this mechanism to downregulate this particular transcript, wouldn't it be more energetically better for the plant not to transcribe it or to degrade it?
Yeah, so I will answer as I would answer to the first year IBMC course. I don't think that the adaptive value of a mechanism is just to save energy. In general. I don't think that energy saving is the motor of natural selection. It might be in some cases. In some of the cases, selection is a tinkerer. It takes what it takes. And
why these genes? Why only these genes? Why is splicing related?
No. What I believe is that this is not, I know this is not the only case in which the abundance of a mature messenger is regulated by RNA processing and not by the rate of transcription. This is well known. And your question is, why is this mechanism operating instead of regulation or degradation of regulation of transcription? I don't know why it is operating. What I know, and even though it's the weakest link of the whole story, is that if I saturate, if I overcome this regulation, I have troubles. But in principle, I wouldn't think that the whole gene regulation must take place at the level of alternative splicing and transcription. But there are many cases now in plants and in animal cells in which the abundance of the mature messenger that is important for selection is not regulated at the level of transcription, but is regulated at the level of splicing or cleavage and polymerization or capping.
Alberto, I asked this same sort of question.
And I probably don't have the same answer.
No, not to you, but to Petri here 2 years ago. And perhaps you have more information now. The light harvesting complex genes are sensitive to the transcription, sensitive to the redox state of the plastoquinone. So I was wondering if there's some effect, if when you looked at this panel, if there was
I don't remember to have seen.
I didn't know so much when I asked.
So the last harvesting complex genes are nuclear genes.
Nuclear genes.
And the proteins
that respond to the redox state of the chloroplast.
No, I have to look at the panel. Maybe Petri didn't know.
He said, he gave me very good answer.
No, I am honest.
But I don't think at that time.
Of the panel? I don't know. Maybe Marcelo, you know if in that panel, you remember? I don't remember if in that panel there was the LHC splicing events. I don't remember. Sorry. What knockout? Knockout of what? Knockout of what, sorry? The RS31? Yeah, the knockout, no, there is no knockout, there is an insertion in it. But it doesn't make the protein. So not making the protein is not as harmful as to make it during the night. That's what we conclude.
Another question here.
Yeah.
You have any idea or hypothesis concerning the nature of the signal that is translocated?
No, we don't have any idea. It's a tough work. For a while, Mika was growing protoplasts, subjecting them to light and dark, collecting the conditioned medium, and using the conditioned medium either on other protoplasts or even on seedlings. And at the beginning, we saw some effect of the conditioned medium of the protoplast exposed to light, but then the effect was lost. And if it's not reproducible after 3 or 4 experiments, I cannot put in risk a thesis, you know. So we have to abandon that. That doesn't mean that in the good conditions, with loss of protoplasts and very well concentrated conditioned medium, we couldn't use that approach to try to identify the signal. But we tried, and as I told you at the beginning, we were very excited because there was an effect, but then it went away. And we tried several times, and then we said stop. So in the case of the shoot to the root, we are. I cannot give you an answer because we don't have the answer yet, but we are suspicious of sugars in terms of some mutants, but it might be again that it's not reproducible, so I have to be sure. But it might be that sugars are involved. Maybe I'll tell you this in a few years. Hola, Eddie.
Hola, ¿cómo estás?
¿Cómo te va?
Bien. Con respecto a la misma pregunta, yo mientras veía tus experimentos me acordaba de unos viejos trabajos de Winslow Preximado y me parece que Diana Madori o algo así, que muestran que hay como fibras ópticas que comunican la luz desde la parte aérea a la raíz. ¿Y podría ser este el caso y de que los eventos estén siendo controlados por otro organismo?
Can I repeat the question in English? Or you can do it. So Edith is asking me, she's recalling an old paper in which there were optic fibers described that communicated light from the shoot to the root. So I wasn't aware of this. I don't know how I could prove that. As Acetabular.
I don't remember. I know it was an open
Okay,
by
but-
Winslow Briggs and
Yeah, but in my naive botany, I would say that the interior of the phloem is very dark. I think the actual phloem and maybe the xylem too, I guess, is very dark. But this is my naive botany, I don't know. But everything is possible, I don't know. It looks like, when you were telling this, I must confess, it surprised me, I was thinking about the new botany that you see in Avatar, in the film, you know? In terms of communications, like neural networks that communicate in the Mother Earth, one plant to the other one, by light. But I think it's great, it's a new, I mean, Avatar is a fabulous movie because it creates a new botany and a new zoology. And this is something that not even Linné there.
More questions? I will do one myself. In regard to the moving signal, so did you think of doing any kind of graph? I mean, with Arabidopsis it's hard to, but maybe other species.
I thought of this, but again, I thought of the Darwin experiments, you know, because Darwin, to study, I think it was phototropism, he did graft no? So I thought of this, but I don't know whether we have the expertise to do this, but it might be. Perhaps we could think it together. So do you know how to do grafts of seedlings or Arabidopsis?
I know there were some papers with Arabidopsis. I don't know how to do it.
Okay.
They're pretty small, but maybe with tobacco.
But those are the old Darwin experiments, that's true. Darwin was incredible because the guy, after talking about natural selection, publishing books and writing fundamental books, he was publishing papers. And he was publishing papers and papers and papers about different things. So it's incredible.
Right. Do you try your experiments between leaves, like shading one part of the shoot?
No, I know what you mean. Trying to identify if I can cover one leaf.
And leave the other ones.
Yeah, no, we haven't tried that. We mainly work with seedlings, not with grown plants. So the number of leaves is limited, but we haven't tried.
Any more questions? Well, we thanks Alberto again.
Thank you.